Okay, so today I was told in advance that the internet in my apartment, they're going to be working on it. So I'm going to record this kind of in advance for you guys so that you have it and that yeah, we can stay on track. So I'm going to ask you to watch the recording for this lecture and then watch another short YouTube video. But today we're going to talk about some terminology that we use in the nervous system and how we come up with these different terms that we use. And then once we define that vocabulary, we're going to talk about the action potential. The other thing I want you to put on your radar again is that you have your first week of nervous system discussion questions, nervous system questions part 1 that are due Friday at midnight. So please make sure you answer those. Okay, diving in here. So unfortunately, the scientists years ago who worked on the central nervous system, the brain and the spinal cord, were different than the scientists who worked on the peripheral nervous system, right? Our nerves that make up our cranial and our spinal nerves, the nerves of your body. And so they came up with different terms that mean similar things, but in the central nervous system versus the peripheral nervous system. So the people working on the central nervous system call a cluster of cell bodies a nuclei. So this is different from the nuclei in your cell that holds the DNA. This is a cluster of cell bodies of neurons, and that is called a nuclei. You might have heard of that term when we're talking about different brain regions. Basolateral nuclei, for example. Nuclei means just a cluster of cell bodies. Now, a ganglia means a small collection of cell bodies, but in the peripheral nervous system. So when we talk about the basal root ganglia coming off of the spinal cord, ganglia there means a small collection of cell bodies in the peripheral nervous system. We have a bundle of neuron processes, usually axons here, that are all traveling together. We call that a tract in the central nervous system. So this is a bunch of axons that travel from one brain region to another. We call that a tract. And that same bundle of neuron processes in the peripheral nervous system we call a nerve. So your spinal nerve is actually not just one single neuron, it's a collection, a bundle of neurons that are all traveling together. We call that a nerve. And finally, we call white matter the dense collection of myelinated processes. These are, again, those axons that are all myelinated to speed up transmission. And gray matter is the unmyelinated processes. Now, the word process or processes, if we're talking about multiple, means axons and dendrites, the things sticking out of the neurons. And so usually gray matter is the dendrites, they're usually unmyelinated, and the cell bodies. So the gray matter is usually like the top half of the neuron, and the white matter would be like the bottom half of the neuron, that axon that's full of myelin. So again, you got to keep these in mind. The best way to study this would be just to make some flashcards of the different terms here. Make sure that you understand which one we use in the central nervous system versus the peripheral nervous system. Okay. Let's use some of those in action. So when we're talking about the afferent and the efferent pathways of the motor, we're looking at motor neurons versus sensory neurons. So in our spinal cord, we have afferent neurons, which come up a nerve. We have efferent neurons that move down a nerve. So afferent meaning towards and efferent meaning away. We will come back to that when we talk about the spinal cord as well. Afferent neurons have their cell bodies outside of the central nervous system. That is where we're taking that sensory information about the world around us. We're seeing things, we're feeling things. That is going to approach the brain and spinal cord via an afferent sensory nerve. And it's going to reach a cell body, and then we'll shoot out the axon up into the spinal cord. Whereas the efferent nerve, the motor neuron, the thing that's going to have the effect on the body, that has a cell body located in the central nervous system, usually in the spinal cord. And a neuron that's fully in the central nervous system, it's a connector between afferent and efferent neurons. We call that an interneuron. So again, more vocab for you to know. Afferent meaning approaching, efferent meaning away. Afferent being our sensory nerve, efferent being our motor nerve. And then an interneuron, neuron that's fully localized in the central nervous system usually acts as a connector between our afferent and our efferent pathways, usually in the spinal cord, as shown in this picture. Okay, so in your nervous system ungraded quiz, you have some fill in the blanks that I want you guys to do about using some of these vocab words. So again, as you go through the lecture, I've posted a bunch of stuff to Canvas to help you kind of engage with the material a little bit. And one of them is an ungraded quiz that would take the place of what we would do in class time. Let's think back to muscle contraction. So when we talked about muscle contraction, we talked about the effect a motor neuron can have on a muscle fiber, on a muscle cell, right? And that motor neuron was going to come from our spinal cord. It was going to release acetylcholine, and acetylcholine was going to bind to that sarcolemma. And acetylcholine we termed a neurotransmitter. Acetylcholine we talked about as a neurotransmitter, and that neurotransmitter had an effect on the membrane of a muscle cell. And that effect was to start an electrical signal. And that electrical signal in the muscle is what caused the muscle to contract. Everyone remember all of those fun hours we spent talking about muscle contraction, right? So I want you to think back to that because a similar process is happening in our neurons. A similar process is happening in our neurons. And that is that electrical signal. And in biology, when we talk about electricity, we talk about the movement of ions. So just like in our muscle cell, where there were ions moving around, creating electricity that caused the muscle to contract, talk about ions moving around in our neuron, that's going to cause that neuron to have a signal, or fire and action potential, just like it did in the muscle. Okay. So neurons, like muscles, have an electrical potential. At rest, the neuron is polarized. It means there's a difference between what the inside is like versus the outside in terms of charge, And again, charge here, we're talking about ions, things like sodium, potassium, not as much chloride and calcium for the neuron. We're mainly talking about sodium and potassium. There is a gradient here. There's a gradient. And that there is going to be negative charge on the inside and positive charge on the outside. Negative charge on the inside, positive charge on the outside. There's going to be a lot of sodium on the outside of our neurons and a lot of potassium on the inside. There's going to be this gradient. In biology, gradients have energy. There is energy associated with the gradients. Gradients can do things because the ions themselves want to move. They want to move across. They want to normalize that gradient, get rid of that gradient. But they can't because remember, our plasma membrane at rest is impermeable to ions. Ions cannot just weasel through that plasma membrane like lipids can, like gases can. So at rest, there is this distribution of charge, this gradient of some key ions, namely sodium and potassium, in our neurons that hold a lot of energy, a lot of potential energy. And the action potential are electrical signals. So your neuron is going to receive a stimuli. And that stimuli can come in a few different forms. When we talk about our senses, we'll talk about the stimuli being light. That is going to be the stimuli from neuron in your eyes that help you see. Sound, that's going to be in your ears, obviously. They're going to help you hear. Pressure, this is all on your skin. You have receptors to pressure. That's the sense of touch that we talk about. Or it can be a chemical signal from a nearby neuron. Again, that chemical signal we call a neurotransmitter. And that neurotransmitter is going to have an effect. And what happens is similar to what happens in the muscles. We're going to open two kinds of channels. So the signal is going to open sodium channels, and sodium is going to rush into our neuron fast. The first thing that happens after we have a stimulus, we have some sort of stimulus, the first thing that happens is sodium channels open, and sodium uses that as an opportunity to try and equalize that gradient. So all of the sodium that's outside the cell is going to rush into the cell because there's less sodium in the cell. The potassium is the second thing that moves. So again, the first thing that moves is sodium through a sodium channel. Then we're going to open up potassium channels. Potassium channels are slower to open. Potassium channels are slower to open. Potassium channels will open, and potassium will leave the cell. This is similar to that idea we talked about in the muscles of a wave of positive coming in and positive coming out. But unlike in the muscles, here in the neuron, they each use their own channel. So it's not just a generic cation channel like it was in the muscles. Here we're talking about two different channels. We're talking about a sodium channel that's separate from a potassium channel. And the sodium channel will open first, and the potassium channel will open second. And keep in mind, we're still talking about that depolarization. So depolarization, again, is a strong change in the membrane of the charge on the membrane, where the membrane becomes positive. So depolarization is a strong change in the membrane, where the membrane, which is usually negative, becomes positive. So if we're looking here at the membrane, we're looking here at the membrane at this top picture, we have the extracellular space. This is the outside. We have the cytosol. This is the inside. The outside is positive. The inside is negative. When we get some sort of stimulus, it's going to open that sodium channel, that voltage-gated sodium channel. Voltage-gated means that what gates the channel, what opens the channel or closes the channel is the voltage, is the difference in charge. So when the membrane becomes positive on the inside, that sodium channel is going to open. And that sodium that's been waiting outside the gates here rushes into the cell. It's super excited. It really wants to get rid of that high sodium gradient. So the sodium is going to rush into our cell. Now, the sodium channel is unique in that it gets tired. It gets tired. It's a tired channel. So after it's open for a short period of time, it will reach a state of inactivation, where this little piece here on the cytosolic side is going to block the channel. Nothing else can happen. Okay? So the sodium channel is normally closed. When we get a depolarization, when we get a change in the voltage of the membrane, when the cell becomes positive on the inside, that sodium channel is going to open, sodium is going to rush into the cell, and then the sodium channel needs a break. It's inactive. And we call that time when the sodium channel is inactive our refractory period. The refractory period is a time when no voltage-gated sodium channels can do anything because they need to rest and recover. So the depolarization is driven by those voltage-gated sodium channels. And that action potential is going to move down the axon. So if we start at time 0 here, okay, the triggering, a stimulus is going to come in. That stimulus is going to change the inside of the membrane to a positive charge, from negative to positive. That change in voltage is what opens our voltage-gated sodium channels. So the first thing is a stimulus changes the voltage at the membrane to be positive on the inside, which is a depolarization. And that depolarization, number 2, opens our voltage-gated sodium channels. And 3, sodium will then rush into the cell. Sodium will rush into the cell, and that will further depolarize the cell. That further depolarization will open nearby sodium channels, And in that way, we're going to move that action potential. We're going to spread that wave of positive charge down the axon with different depolarizations opening more and more sodium channels. And again, what happens behind it is that the sodium channels then become inactivated, shown here in that light blue. So the sodium channels open and then they rest. And that keeps the signal moving in one direction. So the inactivation of our sodium channels, step 3, the inactivation of our sodium channels, keeps the signal from moving backwards. It has to keep moving forward because the channels behind it can't do anything. They need to rest. The second phase of our action potential. So the first phase is that depolarization driven by sodium. That positive charge coming into our axon and moving down the axon is driven by sodium. So number 4 here is a repolarization stage where we need to put the cell back to where it went. We need to bring the positive charge outside the cell and make the inside of the cell more negative. And that is driven by potassium. So potassium channels will open more slowly. Step 4, voltage-gated potassium channels open. And step 5, potassium will leave the cell, returning that membrane to negative on the inside, positive on the outside. Okay? And then what happens in step 6 is that now we need to restore our gradients. Because Now we've got all this potassium outside, all this sodium inside. The cell is all messed up. It prefers to have sodium outside and potassium inside. And so we're going to switch potassium for the sodium using our sodium potassium pump. So this uses ATP, cellular energy, right? Cellular energy currency, to put potassium back on the inside and sodium on the outside. Okay. Now that was a lot. What I want you to do when you watch this video is pause the video right here. I want everyone to pause the video and see if you can write down those 6 steps that I just went through very quickly. Okay, see if you can write down those 6 steps I just went through. Draw yourself a picture. Okay. Now let's talk about what happens when our action potential, when that wave of positive charge reaches the end of the line, when it reaches the axon terminal. So all of this is happening down the axon of a neuron. But what happens when it reaches an axon terminal? Well, our axon terminal is where we're going to convert this to a chemical signal. The axon terminal is where we convert the signal from an electrical signal. The electrical signal is that change in ions. The electrical signal is that change in ions, that wave of sodium coming in followed by potassium going out, that action potential. But that doesn't work at the end of the line because we need to pass that signal to a nearby neuron that's not physically connected. And so the way that we do that is through the release of a chemical called a neurotransmitter. And so what this picture is showing you is it's showing you that the nerve terminals, the axon terminals, are full of what look like little teeny bubbles when you take a picture. So this is an electron microgram picture. This is actually what it looks like if you were to slice up a little bit of neuron and look at the end. So here is that axon terminal, and you can see it's full of bubbles. And these bubbles contain neurotransmitter. These are vesicles. And so the action potential will come down the axon. And when it reaches the terminal, that depolarization is going to open a voltage-gated calcium channel. This is the third ion. So the action potential is driven by sodium and potassium, 1 and 2. But the conversion to a chemical signal is driven by calcium. And this we talked about when we talked about muscle contraction. So the action potential is going to open up our voltage-gated calcium channel, and calcium will rush into the axon terminal. And the calcium is what tells the cell, okay, dump out the neurotransmitter. And the neurotransmitter is going to diffuse along the synaptic cleft here, that gap between 2 neurons, and it will be the stimulus for a nearby neuron to fire their own action potential. Okay. It'll be the stimulus for a nearby neuron to fire their own action potential. And those neurotransmitter will bind to a bunch of receptors in a nearby neuron, triggering that action potential in a nearby neuron, the second neuron. We call that nearby neuron the postsynaptic cell. First cell is our presynaptic cell. It reaches a synapse, a meeting of 2 neurons, and then it becomes our postsynaptic cell. Okay. And again, this is a conversion of an electrical signal to a chemical signal. So that chemical is going to be stored in a vesicle. The vesicle will fuse with the plasma membrane. That will release those neurotransmitters into the synaptic cleft, and the neurotransmitter will bind to a receptor on a postsynaptic cell's membrane. What it will do is it will cause that signal to be propagated, to move to a nearby cell, which will have its own action potential. And all of this happens like this, super fast, right? Just think, snap my fingers, and you can do it in an instant. And that's because of that signal that's traveling all the way from your brain, down your spinal cord, down your hand. And this whole process has to happen in that time in multiple neurons. So think about that when you need to think about the speed at which all of these processes are happening. Okay, what I want you to do is I want you to pause the video at this point and draw a picture. I want you to draw a picture of the action potential, all of the different channels that are involved, okay, and how that releases neurotransmitters. And we're going to stop there for today. So again, we talked about some terminology, and then we talked about the action potential. I spent most of the time on the action potential. And I'm going to link a YouTube video that I think is super important that I want you to watch as well. So I want you to draw some pictures, watch this video, watch a second video, and answer some discussion questions. And we'll pick up with this on Monday.